Hello. This week's lesson is divided into two main categories. One is on why cells divide and how cell division works, and the second one is on human tissues and a little research project that you're going to be doing on it. So cells, if you remember from last week, we talked about what a cell is and how they work. So if you haven't done that lesson yet, I highly recommend you go do that before we do this one. So cell division, why do cells divide? Well, here are four main reasons why we have cell division. And we're going to go through each one of these reasons in a bit of detail, so they're there for you. So it talks about maximum size, we need to grow and reproduce, we need to maintain fresh cells, and we also need to repair. So let's take a look at the first one, size limitations. So we start off with a knife big cell, and it'll eventually divide into two smaller cells. But let's take a look. So the first thing, it goes back to that cell membrane. So the cell membrane is that semi-permeable barrier where things come in and out of the cell. It's the only way for the cell to exchange, let's say, oxygen and carbon dioxide, or for it to exchange nutrients for waste. And as the cell grows, it's going to require more and more resources. So just like you eat more now than you did when you were 3. The larger you get, the more you eat, the more you're going to breathe, and the cell works the same way. And one of the problems, though, is that as the cell grows bigger, its ratio of surface area to volume changes. And so what ends up happening is the distance on the inside of the cell from the barrier, so here's the cell membrane, the distance from the membrane to the inside of the cell, that distance actually grows as well. So it starts becoming too far for things to travel. So let's take a look at a little bit of math here. So let's pretend that you have 3 cubes. One cube that's 1 millimeter by 1 millimeter by 1 millimeter, another one that's 2 millimeters cubed, another one that's 4 millimeters cubed. Well, if you remember back to math, we can do the surface area. So you just take the area of each one of the sides and you multiply it by 6 because it's a cube. So 1 times 1 is 1, times 6 is 6. So this has a surface area of 6 millimeters squared. Now its volume is just that 1 times 1 times 1. So we end up with a volume of 1 millimeter cubed. Now if we remember, the surface area, that cell membrane, is what lets resources in. So for every 1 millimeter squared of surface area, for every 1 millimeter cubed of volume, we have 6 millimeters squared of surface area to provide it. So that's 6 to 1. So that's quite a large ratio. So that means you have lots of surface area to pull in all the resources you need. Now let's just go ahead and double that cube. So now we have a 2 millimeter by 2 millimeter by 2 millimeter cube. Well, we just do the same math, and you'll find that you have a surface area of 24. So look, the surface area got quite a lot larger, right? But here's the problem. When I do volume, the volume also got a lot larger too. So now when I take 24 divided by 8, now I only have a 3 to 1 ratio. That means for every 3 units of surface area, I have 1 unit of volume. Before it was 6 to 1, now it's only 3 to 1, which is actually a lot less. It's half. So now that same surface area has to provide twice as much nutrients to the volume. Okay. Now if I go ahead and double the cube again, 4. Well, I do the math. 96 millimeters of surface area, so a lot of surface area, but look what happened to the volume. The volume went up really big too. It's at 64, and when I divide 96 by 64, I get 1.5 to 1. Well, that's again half of the amount of surface area to volume. Another way to think about it is think of a mall, right? If you have a super huge mall versus, let's say, a little tiny mall. So let's look at maybe going to a strip plaza where you just have a bunch of stores on the outside, and maybe 7 or 8 stores, maybe like a Timmy's on one end and a Pizza Hut or Pizza, and then maybe like a little convenience store and maybe a dollar store, right? Right. And so that would be this little cube. Okay, so nice little mall, right? And then look at a big mall like the Bramley City Center, where it has a couple hundred stores and parking all around the outside. Now, you might be able to get what you want at the Bramley City Center, but think about it this way. Think about if you only had 5 minutes to go shopping. Which one is going to make it easier for you to get to the stuff you need? Well, if I go to the Bramley City Center, I might have to walk like, spend 5 minutes getting parking and then walk like 5-6 minutes to get my item, and by the time I'm done, it's a half an hour to go get one little item. Where the little strip mall, I can just drive up to the store, get what I want, and leave. Okay. Now it's a bit of a kind of not a great, example, but it's kind of the idea that this one is just easier for the nutrients and the waste to kind of be exchanged. Okay, just because you have more surface area than volume. And so that's one of the big reasons, and that's one of the big triggers for division, is like, I'm getting too big, I need to divide. So there you go. Okay, growth and reproduction. Second reason, well, we start as a sperm and egg, and we end up as, at 3 months, this is a little embryo, right? Well, actually a fetus, okay? And then, you have a baby after 9 months, roughly, and then, you guys after, 15, 16 years, and then me after, well, a lot more years than that, right? But we eventually go from that little tiny baby, or little tiny sperm and egg, rather, one cell, to all the way up to a couple trillion cells. Well, the only thing that allows us to do that is by the cells dividing. So if you just took out your calculator and did it, let's see if I can get my calculator up here. You can actually do a little bit of math and see how it grows so fast. And let's just get the calculator up here. And let's just do it. So if I start with one cell, remember I'm just going to be doubling, right? Times 2. It goes to 4. Times 2 again. See, every time I multiply it by 2, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So after doubling 15 times, I'm already up to 32,000 cells. And then let's say 1, 2, 3, 4, 5, 6. Well, now I'm up to, what's that? Over 2 million, And eventually it grows up into the billions and trillions really fast, right? So now what are we at? We're at about 2.5 trillion or 2 trillion or so cells, which is kind of in the ballpark of you. And I only had to push the button about 25, 30 times. Okay, so you can go from 1 to trillions really, really, really fast. And that's what happens. It happens in 9 months. Okay, so there you go. Growth and reproduction. So without it, without cell division, we couldn't have people, and people wouldn't grow. All right, next one. Maintenance. Okay, this is different than repair. We haven't caused damage. These things naturally wear out over time, or maybe we need to get bigger. I don't know how you feel about snakes. I like snakes. Obviously, I don't want to pet a poisonous one, but generally speaking, they're not so bad. Every hour you have a billion cells that die, but you also have a billion cells that are created. And in fact, you being teenagers, you might have a billion cells that are dying, and you have just a slightly more than a billion cells being created, so that's how you're growing, right? Where me, I'm older, I'm not growing anymore, so it's about the same number, right? So in order for this to happen, you need to be continually replacing cells. So it's kind of a scary idea to think of every day you're losing 24 billion cells. But don't worry, you're growing that many cells too, so you're not dying. It's okay. Well, the snake, for example, that's what he does. He grows a new layer of skin underneath the old skin, and then when that new layer is ready, he basically just scratches off and rubs it off against rocks, and the old skin just falls off. In fact, if you ever go in an area where there are snakes, you'll find these they kind of look like, crusty tubes on the ground, and that's actually snake skin. And there you go. So that's them. I mean, us, instead of having our skin all fall off as one giant tube here, we have our skin just slowly fall off over time. And that's in fact what most of the dust in your house is actually just dead skin. So there you go. We're always creating and losing cells. Think of your hair, right? You have lots of hair come off when you shower, or guys, if you shave yet, or even just the facial hair, it falls out. You're creating cells to make that, and then it's always being regenerated. Okay, there you go. But that's just maintenance. That's just normal wear and tear. That's not injury or anything. That's just regular maintenance of it. Very few of the cells, if any of the cells that you're born with, you die with. So even like your brain cells and stuff, they do kind of reproduce over time. So there we go. Next one, repair. Okay, so we've all been injured, right? So we took this little newt. Now, I didn't do it. Somebody did it in a lab. I know they do these kind of things. Kind of cruel, but they knew what was going to happen with the newt, so they weren't worried. And they took his arm, his little hand, and they cut it off. And you see after about a week, it starts growing a little nub. And then after a little longer, you start seeing the hand come out. And after, a couple months here, he grows his arm back. Okay, and there's other creatures out there that can regrow limb parts. Now we can't do that. If we lose like a big chunk of something, it never really comes back. But think about like when you get a cut in your hand, right? Let's say you just get like a little cut, you know, not too deep, just bleeds a little bit, but you don't need stitches. You know, it takes 4 to 5, 4 weeks or so, and it goes away. You don't even see it anymore. Well, that's not because the skin healed. It's not because that skin went back together and fixed itself. It's because new skin grew to replace it. And that's what repair is for us. It's not that we fix the stuff that is damaged. We grow new stuff to replace the old stuff. And that all happens through cell division. Same thing if you get a burn, you know, you burn your tongue. You know, your tongue's going to be a little bit sore for a few days. Well, that's how long it takes for those cells to reproduce. In fact, the cells inside your mouth, like your cheek and your tongue, are actually some of the faster growing cells in your body because they get damaged a lot, right? So we're repairing it. And there you go. So we need it for repairing. Okay, and so the cell cycle, we got to mention this a little bit here, and we'll get more into detail in the next presentation. But here we go. Just like people, cells are created, they live, they die. From the time they're created to the time they die, we call that the cell cycle. Okay, in humans, different kinds of cells have different sort of timelines to do it. The average cell is 14 to 28 days, so 2 to 4 weeks. Now we have some that are a lot quicker. Okay, and we have some that are a lot slower. In fact, red blood cells, it says here, 120 days. That's about 4 months. And if you think about it, if you're training for a marathon, or think of like a fighter training for a UFC match, they're going to train for 4 months because they need to make sure all the red blood cells in their body are good and strong, and so they have to wait till all the old ones die off and are replaced with good, strong ones. Okay. Same thing if you like to do weights in the gym. It's going to take you a few months for you to really notice some difference, right? Because it takes that long for your muscle cells to repair. Maybe not 120 days, but it still takes a few days. But like I said, with the cells in your mouth, right? If you get a little cut in your mouth or burn your tongue, it's definitely less than 2 weeks. And maybe I burnt my tongue about 3 days ago. It's still a little bit there, but it's almost gone, right? So it goes a lot quicker. So during most of the cell cycle, the cell is preparing to divide. In fact, the cell division only happens for a very, very small time of the life of the cell. Most of the time, the cell is just hanging out and growing. Okay, and we'll talk about that. Now, mitosis is just when we take one cell and create two. Exact copy, duplicate. That's what we're going to cover, this word called mitosis. There is another process out there called meiosis, which talks about how we produce sperm and egg. Okay, but in this presentation, when we talk about growth and repair, we're talking about just mitosis, okay? And there you go. So let's end it here, and we'll get into this rest of the stuff in the next presentation. Okay. So what this leads us into is something called cell division and mitosis. Now, what you proper way to say it is mitosis. So it's like you hurt your toe and then your sister says, "What's wrong?" And you say mitosis, right? Okay, that's it. Mitosis. There we go. Mitosis. So the first question I got for you is, what determines if you're a girl or a boy? Now again, I'm talking about sex, not gender. So quick little discussion about that. Your sex is biology. It's we'll mention that in a second. We'll talk about what the biology says. Gender is a choice, so it's whatever you prefer to identify as. So we have people out there that might have a sex of a boy or a girl, but they choose to identify as something else, which, hey, no problem. That's good. It's good that you choose to live your life the way that makes you happy, right? But in terms of biology, there is a correct answer for sex, not gender. And that's why they put on question forms instead of sex. They often put gender because it's what you identify as. Or if you go to the doctor's going to want to know both, right? Nonetheless, so what determines if you're a boy or a girl? Well, it's your genes. So it's your genetics. Okay? And that's really what determines it. So if we remember, we had the nucleus. It's the control center of the cell, and it decides what kind of cell the cell will be. Okay? It will also decide what the function of the cell will perform. It will also determine when it dies. Okay? And here's an example. These are nerve cells. Okay? And so it's the nucleus, which you can actually see on the inside, that tells it to grow these nice long arms and tells it to do all the things that it does. And so that cell looks that way because of the nucleus, the control center. Now, what's inside the nucleus? Well, it's that good old stuff called DNA, which is also called deoxyribonucleic acid. Okay? And DNA is the blueprint, the instructions. Okay? It's what the nucleus reads to instruct the cell what to do. And let's get into looking what DNA looks like. So here's DNA. And it is a very long molecule. In fact, they're meters long. You know, even though they're microscopic, they're very, very long. If we undo them, they look like a twisted ladder or like a spiral staircase. Okay, they're first discovered in 1953. Now, I'm not going to mention who discovered it because even though they claimed to have discovered it, they actually stole the work from a woman and claimed it as their own, and weren't discovered that they made that mistake until a long time ago, or until a long time after. Go ahead, you can look it up on YouTube. There's lots of interesting videos about it, but it's somewhat in debate right now. So, but nonetheless, it was in the 50s that it was discovered, and we noticed that it's the same in every organism. So every single living thing on Earth has DNA, and they are structured almost the same. Okay, and in fact, the DNA in like a little tiny plant is actually very similar to your DNA. You actually have very, very little differences. And it's kind of amazing that the little tiny differences make you into a human being rather than a tree. So there we go. And DNA is the basis for all life, and it's you can think of it as the computer program. Okay, it's the code, bunch of zeros and ones. Except this one doesn't use zeros and ones. It has 4 different components to it. And the most important part, and why we're going to look at mitosis, is because it talks about how we split it during cell division, okay, so that we can make exact copies of itself. All right. So let's take a look at how that does. So DNA molecules, it's like a giant string, but if you twist a string, or if you have your earbuds, right? You have your earbuds; they're nice and strong, or sorry, nice and thin and long, but let's say they're in your pocket for too long, they end up coiling up. Even though it's one string, it ends up looking like a ball, right? Well, that's the same thing DNA does. Okay, it's without the folding, each strand would be about 2 meters long, right? 6 feet, actually the perfect distance to stay away from people right now. But really, what it is it's kind of like a ball of string, kind of like what we have in this bucket, or this is an actual. I think this is an electron microscope view of it, but nonetheless, it's a nice giant long string that's been curled into a ball. Okay. Now, what you have in your genes is a gene is a very short section of that very long DNA molecule. So here's a short section, and in that little section is what's called a gene, with a very specific code. Now, almost all traits in you are controlled by more than one gene, but for simplicity's sake, we'll just talk about certain genes, right? So examples would be like your eye color, your height, your hair color. Other things could be what shape your nose is going to be, the shape of your toes, all kinds of things, right? Are you going to be diabetic, right? These are all things that are kind of built into your DNA. If you're going to be a boy or a girl, right? If your sex is boy or girl, that's all in your code, right? Now each gene has each DNA molecule has about 1,000 genes on it. Now, it's not to say you only have 1,000 genes, because you have more than one strand of DNA. Okay. So there you go. So those are genes. Genes are just sections of the DNA that have codes for it. And think of your computer. You might have like 100 programs on your computer. Each one has its own little section of code, but it's on one hard drive. So it's the same sort of example here. So what we have instead are chromosomes. They're made up of a single strand of DNA. They've gotten kind of twisted and folded, and so they have a specific shape. That's the shape of an X. Everybody has an X. And each plant and animal have a specific number of chromosomes. Well, I think you can't have, animals or whatnot that both have the same number, but, humans, we all have the same number as each other, generally speaking. There are a few exceptions, but humans, we have 46 chromosomes, and they're divided into 2 categories. We have 22 pairs of just regular chromosomes. Now each one of those has different genes in them. Okay, so it's not like we have 22 sets of the same genes. So we have 22 chromosomes, and each have genes on them. And then we have two what we call sex chromosomes. And I've shown you one here, which is the X. Now you have two, so everybody has an X. But if you have two X's, that makes you female. If you have an X and a Y, that actually makes you male. And that's it. Out of your 46 chromosomes, it's only one that's different in terms of making you, sex-wise, a boy or a girl. And there you go. So you'd have your chromosome, and then the genes are like little tiny parts of it. So here we go. There's the egg, there's the sperm, there's our chromosomes going together. In humans, your mother gives you 23 chromosomes, and your father gives you 23 chromosomes to give you 46 chromosomes to develop the new fetus. There's 23 chromosomes in the egg, there's 23 chromosomes in the sperm. So that means every cell has 46 chromosomes. And there you go. So let's get a little bit more into what this looks like. So here's your nucleus, and inside the nucleus you have these chromosomes floating around. If I took a chromosome and I unwound it, it's like a ball of yarn, you'd eventually get a strand of DNA, and then that strand of DNA contains genes on it. Now, I told you before there's the cell cycle. Well, there's 3 stages of the cell cycle. So the first stage is interphase, which is actually this giant stage here. In fact, this visual doesn't even represent it that well, because it actually would be like, for some cells, it would be like 99% of this wheel. Okay, but we just made it 3/4 here. But think, man, your cell only divides once every like 4 weeks, and it doesn't take very long to do that. So interphase should actually be much larger, but this is just a diagram. The second phase is mitosis, which is this whole stage where we go through division. So we start with 1 cell, we end up with 2 cells, and that's called the mitosis phase. And then the end of the mitosis is what we call cytokinesis, which is where we actually split the cell. Okay. So you have that one big cell, and it gets split into 2 cells. And the moment that happens, we call that cytokinesis. And that's the end of the mitotic phase. Okay, and mitosis is splitting, mitotic is the phase. Sorry, I should have made it clear here. And then we go back into the cell cycle, back into interphase. So let's look at stages 2 to 6, right? So this is stage 1, and then we have 2, 3, 4, 5, and 6. Stage 2 to 6, this is the mitotic phase. Well, it's the process of dividing the DNA, or the nuclear material, and there's 4 stages in mitosis. Okay, prophase, metaphase, anaphase, and telophase. And we're just going to go through those and show some pictures here. So stage 1, the cell grows. Right, so interphase. The cell grows and it starts duplicating its DNA. It's making copies of its DNA, because when you split, you want both cells to have their own copy. Okay. It's like you're doing some group work, or maybe you're doing a lab together and one person's writing down notes, but before the end of the period you'd want to copy down the notes so you both have it. So you got to make a copy before you split, right? Before you leave. So it's the same idea. But the DNA hasn't become swollen yet. Like, it hasn't kind of clumped up really big, so you still can't see it, but it's duplicating. Alright, and that's all this happens in interphase. We're just going from a little tiny cell to a big cell. Alright, and then this would be a diagram that would kind of show it. We talked about, when we looked at cell diagrams, the centrioles, and these ones will become very important as we move on. Nuclear envelope, so that's like the membrane for the nucleus. We have the nucleolus, which is, yeah, there we go. So don't want to get too much into that, but we're just basically growing here. Stage 2, prophase. So now we're getting into mitosis, right? The nucleus dissolves, so this membrane here goes away, which allows the DNA to condense and coil into visible chromosomes. Okay, and the chromosomes are identical pairs attached to each other. Okay, so you have 2 chromosomes. Like, think of little firecrackers you would, pull at New Year's, where you each grab a side, or turkey wishbone, and then they're kind of stuck together. You each have a side, but they're stuck together. So they've kind of stuck together, but they are duplicated. There are pairs. And that's all happens in prophase. And we just things become visible and the nuclear membrane dissolves. Now we have metaphase, which the chromosomes are pushed together in the middle by central spindle fibers. Okay, by these fibers we call spindle fibers. Now here we have the centrioles, and then you got these spindle fibers that are attached. They're just like little wires. Think of them as like almost coiled springs, or like a screw that when you turn it, it extends, and then you turn it the other way and it contracts. It's like a screw, right? You turn it to the right and it will go further into the wood, and you turn it to the left and it'll come out of the wood. And that's how these spindle fibers work. That's why we call them spindle, because they turn. And here's a picture. So what happens is you have your centrioles here, and then you got the spindle fibers coming out, grabbing onto all the chromosomes. And remember, they're lined up. They now have lined up in the middle. And right, there's one copy and there's another copy, right? And it doesn't matter which way they flip, so that's why this kind of shows different colors on both sides. But this is one copy and that's another copy, okay? Don't think of it as a copy being this way, think of it as a copy being this way. And then this is just an imaginary line, and things actually line up almost perfectly in the center. See? They line up almost perfectly in the center here. Okay? And so we've just kind of had a little anchor on each end and these little fibers that reach out and grab it, and they've all lined up in the middle. Okay. So that's metaphase, kind of a lot going on here. Now what happens in anaphase is we start turning these spindle fibers, which pull apart the chromosomes, and they're held a lot tighter to the spindle fibers than to each other. So when you turn those spindle fibers and start pulling them back towards their own end, it separates the chromosomes from each other, so they're no longer next to their identical pair, and one copy of each ends up coming to either end. So like, let's just take this outside one. See, this was attached to here, but when I pull them, they snap off in the middle, so that you have one copy going this way, and you got one copy going this way. Okay, and that's what's happening in anaphase. Okay, we're just using those spindle fibers to pull them. Now, think of like chewing gum. When you pull chewing gum, or even like, let's say, what do they call it? Bread dough, right? It doesn't stay thick throughout the whole thing. It'll start thinning out in the middle. Well, so in anaphase, we're pulling it. What happens in telophase is as we're pulling it, it starts kind of folding in the middle because we're pulling things towards the end. And then we end up having the chromosomes all getting pulled back to where those centrioles are, and then the cell's like, hey, I'm going to go put a nuclear membrane back around it. So we end up getting our nucleus reformed, or nuclei reformed, and then they start, DNAs go from being the chromosomes and they unfold back into chromatin again. So now what I have is a cell with two nuclei on opposite sides, and these walls that start coming in here. And this picture shows it a bit better. So now they're starting to kind of thin out in the middle, and we have nucleus on other sides. Now all the other organelles, like you have lots of mitochondria, lots of these, and they're just in the cytoplasm, so they're already kind of divided evenly. And plus, if you have a nucleus, you can just make more, so it's not a problem. It's just really the nucleus that we need to make sure gets separated properly, because it has all the DNA in it. So there we go, and now it's starting to squeeze through the middle here. And once this side touches this side, it'll be a new cell, two cells. Okay, so that's telophase, and then cytokinesis is when it finally closes off, and boom, I got two new cells. I've pinched it. And then after cytokinesis, see I have two smaller cells, right? What does it do? Well, it'll sit in interphase, it'll grow. Once it grows big enough, it'll go back through prophase, then it'll go to metaphase, telophase. Well, I missed anaphase in there. I scrolled through anaphase, telophase, and then back to cytokinesis. And it just keeps doing this. So each one of these will turn into two cells. So then I'll have 4, and then I'll have 8, and then I'll have 16, and 32, and et cetera, and that's how we grow. Yeah, there you go. So if you have questions about this cycle, I really strongly suggest you ask me. If not, there's a lot of really good simulations out there, which I'm going to show you one in a minute here, a couple minutes. But yeah. Now, something we like to do that's a bit fun is we got to find some ways to remember the steps. Now, it's in your course not so important. You can write them down and have it, but it's always an interesting way. And in fact, this is one of the things I struggled with in school, and it actually wasn't until I started teaching it that I was like, oh man, I wish I would have known how to do this. It was so easy. So if I just line out the letters, right? Interphase, prophase, metaphase, anaphase, telophase, cytokinesis, right? Well, I could just make like little word games, right? Like I pet monkeys at the circus, or I play music at the concert. Now, my favorite one, and I think a student came up with this 6, 7 years ago, is Indian people make amazing tandoori chicken. And that's the one I remember. Indian people make amazing tandoori chicken. Right? Interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Indian people make amazing tandoori chicken. And that's just a little kind of funny way of remembering the cycle. Right? And you can try making your own. So what we want to look at now is a little animation, which I've already opened. And it will just take us through the cell cycle. So I'm just going to click through it instead. Okay, so there we have interphase. The cell is just kind of growing. And you see here, this is the actual microscope imaging of it. So, and this is just an animation to make it easier to see. So there's our interphase. It's just kind of sitting around not doing much. And there's the prophase. Okay, so things start lining up. And then, well, prometaphase is kind of a stop between prophase and metaphase. But let's go to metaphase. See, things are nice and lined up. And we can see the imaging here. Things line up in the middle. And then what's going to happen next? Well, remember those spindle fibers are going to start kind of screwing and turning and snap the identical chromosomes apart and pull them to their sides. So that's what we see happening in anaphase. Okay, telophase is going to group them on their ends, and then now we can start plopping a cell membrane around them, right? So then we have cytokinesis, where you see here the cytoplasm is sealed off. Well, it's about to be sealed off with cell membranes, right? It's going to be completely pinched off, and once it does that, we have 2 new cells, and then each one of those cells can go back into interphase. So I'll actually just play this for you. And there it is happening live. And that's what your cells are doing, well, your body is doing 1 billion times an hour. And that's about all I have for this. So this video is, what, 32 minutes? So you've gone through that about 500 million times since you started listening to this. Well, that's it about cell division and mitosis. And then what you want to do is just make sure you understand the stuff in this video, and then check out the tissues presentation, which will be much, much shorter, which will be right after this. Thanks. Bye.